Hello everyone, I'm Babita, your instructor for today. Yesterday we studied about monohybrid and dihybrid cross. Today we are going to study test cross and back cross. Test cross is a very important cross in genetics. Let us see what this test cross is. Test cross is generally used to find out the unknown. What does it mean? Let me change the color first. What does it mean to find out the unknown? When we see in a monohybrid cross, the different types of genes that we had seen were capital T, small t, and capital T, small t. So, this is genetic makeup of a plant. And if we do not know, we cannot see this one. Phenotype is morphological, we can see and say whether the plant is tall or dwarf. But how do we come to know of genetic makeup? If we are given tall plant, so whether it is having capital T gene or capital T small t gene, because both of them represent will represent phenotypically tall plant. So the plant that we are given, whether it has capital T gene or capital T small t gene, this we can find out with the help of test cross. So what do we do in this case? We take capital T and cross it with small t, or this is one, another is capital T small t and cross it with same homozygous recessive plant. What will happen? This we have studied earlier also many times. So what will happen in this case? Gamete, only one type of gamete will be formed. Only one type of gamete will be formed. So all the F1 will be tall. In this case, two types of gametes will be formed. In this case, only one type of gamete will be formed. So F1 we have capital T small t and small t small t. So tall and dwarf are formed in the ratio of 1:1. So in case of monohybrid cross, we have all the plants which are in this, when we have the genetic makeup as capital T, all the plants that will be produced will be tall type. But if we have genotype capital T small t, the plants that will be produced will be in the ratio of 1:1. The number of tall plants will be equal to number of dwarf plants, and hence we will get the ratio as 1:1. So if we are getting the ratio as 1:1, that means with the genotype of the tall plant was capital T small t or heterozygous. And if we get all the same type of plant, that is all the plants are tall, that means the genotype of the tall plant was homozygous for tall. So this is how we can find out, if tall plant is given, we can find out what was the genotype of the plant by simply crossing it with homozygous recessive plant. Next, what is back cross? Back cross is generally done to improve the breed. Here what we do, we take a plant, suppose we have a plant. We if we want to improve breed of any of the plants, what we will do, we will cross it with capital T, capital T. So this is already capital T. So what will happen? Only one type of gamete is produced. All the plant that is produced is tall. In this case, what will happen? Because it is homozygous, only one type of gamete is produced. Only one type of gamete is produced, so all the plant is tall. We had started with recessive, but when we crossed it with homozygous dominant, we got all the plant as tall. So we got dwarf. From dwarf plant we have moved to tall plant. This is how it is improving that quality of the plant, and then this will again be crossed with small t, capital T, and further improvement in plant will take place. And finally we can get this type of plant by continuously breeding it with capital T, capital T. What will happen in this case? We have two gametes here, only one in this case. So 50%, though all the plants are tall, but we have 50% of this type and 50% of heterozygous type. If this is again crossed, so we will keep on getting tall, 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 and ultimately after 5 subsequent generation we will get the pure breed. Now let us see test cross in dihybrid cross. If we consider a dihybrid cross, what will happen? So it can be capital T, capital T, capital Y, capital Y into small t, small t, small y, small y. And the second can be capital T, small t, capital Y, small y into small t, small t, small y, small y. How many types of gamete will be produced in this case? Capital T, because only homozygous genes are there, so we have only one type of gamete, and hence all the plants will be tall, having yellow seeds. All the plants will be tall, having yellow seeds. What will happen in this case? Because this has two heterozygous genes, so we are going to have 4 gametes. One is capital T, capital Y, capital T, small t, capital Y, small t, small y. This will have only one type of gamete, that is small t, small y. So if we combine this with the gametes that we have, capital T, small t, capital Y, small y, what is the phenotype? Tall with yellow seed. For this one, what will be the phenotype? Capital T, small y, small y. Tall with green seed. If we take this one, small t, capital Y, small y. This will be dwarf with yellow seed. What about this one? Small t, small y, small y. This will be dwarf with green seed. So what is the ratio of all the different types of phenotypes? 1 is to 1 is to 1 is to 1. So if we get 1 is to 1 is to 1 is to 1 is 1 ratio, that means the plant that we had taken had heterozygous genetic makeup. Whereas if we get only one type of plant, that means the plant that we had taken was of homozygous gene having dominant trait. So this is how we can test what kind of genetic makeup was present in the plant without doing the experiments for this. Morphologically, we can see the results and find out what might have been the genetic makeup of the plant that we had taken into consideration. So this is all about test cross and back cross. There is another cross called reciprocal cross, though it is not used much. What happens in reciprocal cross? Suppose we have two plants A and B, and we take male gamete from this plant and female gamete from this plant, and they are reproduced. In case of reciprocal means opposite. So in another, when we do another cross, we will take female gamete from A and male gamete from B. So we will get another generation. So we are using the same plant for getting male gamete and female gamete both. But if we use male gamete from one plant, female gamete from other plant, and for second cross, if we take female gamete from the first plant and male gamete from the other plant, it is called reciprocal cross. So this is all about today's session. You have to specially remember the test cross. Test cross is a very common question that comes in examinations. So thank you for watching. Happy learning. Do subscribe to get notification for when the video is uploaded. If you like it, do share it with your friends. Thank you once again.